Hello, good morning. Today we discuss the part 3 control release drug delivery system, in which we discuss the release mechanism of controlled drug delivery system or sustained drug delivery system, design and development of oral sustained release system is divided into two parts. That is, part A, the continuous release system. This system releases the drug continuously over an extended period of time. And B part is the pulsatile release system. These are characterized by a time period of no release followed by rapid and complete or extended drug release. Again, the pulsatile drug delivery system is again divided into two. A single pulse system, there are rupturable dosage form that completely release the drug after a time lag. And second is multiple pulse system. These are those that behave like continuous release controlled drug delivery system after a specific time lag. Let's we discuss the release mechanism of controlled drug delivery system or different approach. Depending upon the manner of drug release, this system are classified basically two approach: physicochemical approach and biological approach. In physicochemical approach, it is again divided as a dissolution control release, diffusion control release, dissolution and diffusion control release, ion exchange, resin drug complex, osmotic pressure control system, pH independent formulations, alter density based control release or sustained release formulation. And in case of biological approach, again it is divided into delayed transient or continuous release system, that is pulsatile drug delivery system, biopolymer based sustained drug delivery system, and gastro-retentive based sustained release formulation. One by one we are discussing. In case of physical chemical approach of sustained release drug delivery system, the continuous release system, here the first one is the dissolution control release system, can be obtained by slowing the dissolution rate of the drug in the GI mucin or incorporating drug in an insoluble polymer or coating the drug particle or granules with the polymeric material of varying the thickness. Here, the rate limiting step of dissolution of the drug is the dissolution across the aqueous boundary layer. The rate of dissolution can be approximated by equation dm by dt is equal to ads divided by h. Here S is the aqueous solubility of drug, A is the surface area of the dissolving particle or tablet, D is the diffusibility of the drug, and H is the thickness of the boundary layer. The dissolution system is of three types: the matrix dissolution control system, reservoir controlled dissolution control system, reservoir dissolution control system, cube root dissolution equation. Is a matrix system in which the matrix system are also called as a monolithic, in which the drug is homogeneously dispersed throughout the rate controlling medium. Here rate controlling polymer are the beeswax, carnauba wax and hydrogenous castor oil. And second is the reservoir system. Here The drug particles are coated or encapsulated by one of the several microencapsulation techniques which slowly dissolve the material over like cellulose, PEG, polyethylene and polymethylcrylate and wax, etc. Here the drug, another mechanism is the cube root dissolution equation. Higson and Crown cube root law of the dissolution is used. Here Wg is the original mass of the drug, that is power is 1/3 minus W, the power is 1/3, that means the cube root of original mass of the drug minus cube root of mass of the drug remaining to dissolve at the time t is equal to kT. Is the K is the dissolution rate constant and W is the mass of drug remaining to dissolve at time t. If you see the matrix tablet, it is at the surface side, that is erosion is occurs, swelling, and then the surface particle are dissolved. So first mechanism is dissolution, and after that the central one particle are diffused out. So the second mechanism is diffusion, and the here all the matrix polymer are erode. So last step is erosion. So if you see the matrix tablet, the first one, the expansion, then diffusion and erosion. Second is the diffusion control release system. In that, the diffusion of dissolved drug through the polymeric barrier is a rate limiting step. The drug release rate is never zero order, since the diffuse path length increase with time as the insoluble matrix is gradually depleted of drug. The drug release can also be calculated by the equation dm by dt is equal to adk into delta C divided by L. Is the area, D is the diffusion coefficient, K is the partition coefficient, 1 L is the diffusion path length, and delta C the concentration difference across the membrane. Here again the example of the diffusion control system is the matrix diffusion control system and reservoir. Here the matrix diffusion control system, they are the polymer which is used to prepare the matrix is the guar gum, tragacan, that is hydrophilic gum, synthetic is polyacrylamide, and semi-synthetic is hydroxypropyl methyl cellulose, carnauba wax, and methyl cellulose. The movement of the drug molecule from the region of higher concentration to one of the lower concentration. In case of matrix system, the drug is dispersed in the insoluble matrix of rigid non-soluble hydrophobic material or soluble hydrophobic substance. In case of reservoir system, the drug is at the center one, and outer is the polymer, which is either the rigid or semi-permeable or erodible. The system are hollow, containing inner core of the drug surrounded in a water insoluble polymer membrane. The third one, and so about two one in this example is matrix device and reservoir device, right? So in case of dissolution and diffusion control release system, in such system the drug core is encased in the particularly soluble membrane. The pore are thus created due to the dissolution of part of the membrane which permit the entry of the aqueous medium into the core, and hence drug dissolve and allow diffuse of the dissolved drug out of the system. Here that is possible in osmotic drug delivery system. So in that case, the both dissolution and diffusion both are possible. The method of using the ion exchange, it is based on the drug resin complex formulation. When the ionic solution is kept in contact with the ionic resin, the drug from those complex gets exchanged in the gastrointestinal tracts and thus released with excess of the sodium chloride present in gastrointestinal tract. So here the ion is goes into the device and the drug is released out. That is exchange of ion, and that's why the drug is released out from the device, right? Fifth one is the method using the osmotic pressure. In this system, the flow of liquid into the release unit uh, driven by the difference in the osmotic pressure between the inside and the outside of the release unit is used as the rate controlling system. Here the basically the osmotic pressure play a role, and here the mechanism which is working is the osmosis. Here the mechanism which is working in the osmosis, right? That is, we are discussed detail in osmotic chapter. pH independent formulation. The buffer formulation, a buffer controlled release formulation is prepared by mixing a basic or acidic drug with one or more buffering agent, the salt of phosphoric, citric and tartaric acid, granulating with the appropriate pharmaceutical excipient and coating with GI fluid permeable film forming polymer. When the GI fluid permeates through the membrane, the buffering agent adjusts the fluid inside the suitable constant pH, thereby rendering a constant rate of drug or release. The next one is the seventh one is alternate density based sustained release formulation. It is reasonable to expect that unless a delivery system remain in the vicinity of the absorption site until most, if not all, its drug content is released, it would have limited utility. To this end, several approaches have been developed to prolong the residence time of drug delivery system in the GIT. Is the high density approach, low density approach, right? So here we alter the density, and because of altering the density, we increase the residence time, so the drug remain more time at the absorption area, and that's why the absorption is increased. We discussed the biological approach, in which the first one is delay transist and continuous release system. Those drug delivery system deliver the drug at a specific time as per biological or pathological need of the disease, resulting in improved patient therapeutic efficacy and compliance. This drug release is controlled by the stimuli like the biological pH enzyme present in the body or drug delivery system. The biopolymer based SR or CR formulation. Here the bio nanoparticles in micron size range, that is 15 to 1000 nanometer, made from the biological and physiological lipid which are biodegradable, are used as a colloidal drug carrier for IV application. At room temperature, the particles are in the solid state. Therefore, the mobility of the incorporated drug is reduced, which is prerequisite for controlled release system. The third one is the gastro-retention based sustained release formulation. The gastro-retention dosage form are the drug delivery formulation that are designed to be retained in a stomach for a longer period of time, and the release their active material, and thereby enhance the sustained and prolonged input of the drug to the support part of the gastrointestinal tract, right? Yeah, the pH, the stomach pH are we are utilized for the mechanism in which the device is remain in stomach for longer period of time. Okay, we will discuss the terminology next time.